Whenever we start working with the isolation of the DNA, the main concern is about its purification. As the DNA has to be isolated from the cells after digesting or after damaging the cells. As a result of this damage, there are a number of unwanted materials, particles, cell organelles which comes in the solution. So we have to purify, or we have to take our DNA from out of those unwanted molecules. A cell extract mixture contains DNA, RNA, proteins, cellular organelles, membranes, and other molecules which might have been stored in the cell. In biotechnology, the organic extraction method is quite common to purify the DNA. For that purpose, Phenolchloroform method is quite commonly and routinely used in the lab because of the property of phenolchloroform that it denatures the proteins. On the other hand, the nucleic acid, that is DNA and RNA, they remain in the aqueous layer. That is the reason we have to perform the organic extraction so that we can remove major chunk of proteins from the cell extract. This picture shows you the cell extracts is prepared, phenol is added to this cell extract, and the layers have been separated after centrifugation. The bottom layer is of phenol, the aqueous layer consisting of the nucleic acid, DNA and RNA, whereas you can see that the proteins, they are going to coagulate at the interface of the phenol and the aqueous layer. Once we separate the nucleic acid or DNA from the proteins and other cellular debris, the purification needs ethanol precipitation. Ethanol is quite commonly used method for DNA precipitation. By this method, we can precipitate the DNA from any sample, from any volume of the sample, and concentration can be increased of the DNA. By ethanol precipitation, another benefit is to remove the traces of phenol, chloroform, or any unincorporated nucleotides or oligonucleotide primers, or anything which is unwanted during further use of DNA. Once DNA is purified using ethanol precipitation method, we can use it for further processing.